I'll just show you the learning objectives for today's session. We have to, by the end of this session, you should be able to differentiate between different pathways of complement activation and their regulation, discuss the role of complement proteins in dealing with pathogens and immune complexes, describe the laboratory and clinical significance of complement system. And how the whole thing will be discussed. We'll first talk about what is complement system, then the complement cascade and its activation, how that activation take place, because normally the complement system is not active all the time. Then once the complement system is activated, what are its functions? So why we need to activate the complement system. Then the role of complement protein in extra and intravascular hemolysis. So that's the clinical aspect of complement protein that we'll be discussing. And another thing that we need to know is, once it is activated, what are the components that will keep it in control? So the regulation part, and finally the deficiencies that occur, why it occur, in which clinical conditions, and the detection of these proteins as well. So first thing first, what is complement system? It's a protein like the different proteins in the body, and it makes about 10% of your serum protein. So if you measure, like there is albumin in the blood, these proteins are also in the serum, and they are basically globulins, beta globulins. And like most of the proteins, they are produced by the liver. These proteins are also produced by the liver, but it's not the only source that produces complement proteins. It is also produced by the monocytes, macrophages, and the epithelial cells of the GI tract and genitourinary tract. So these are the sources of the complement proteins. And there are about more than 40 different proteins that are involved in this system. As I earlier said, that complement proteins are not active all the time. They are active, and then they are inactive normally. They have to be activated when the pathogen or the immune complexes are in your body, and after that they need to be inactivated, that they don't cause damage to your body all the time. So there are three main pathways that activate the complement proteins. One is called the classical pathway, the other one is called as the alternative pathway, and the third one is called as the mannose binding lectin pathway. So these are the three pathways that activate the complement proteins. I'll show you, one by one the steps how it is activated. So you have to remember that these three pathways have got three major components. So number one is how it is initiated, how it is starting, whether it is the classical pathway, whether it is the mannose binding pathway or the alternative pathway, what triggers these pathways. Number two, the middle component of these pathway is C3 convertase. We'll come to that in a moment. And finally, the membrane attack complex is formed. So that's the final outcome or the end product of these pathways. So if you look at these boxes, the classical pathway, it is initiated by antigen-antibody, complex. So I told you earlier in my lecture that there are 5 immunoglobulins in the body: GAME-D, IgG, IgA, IgM, IgD, and IgE. These classical pathways are only initiated or activated by IgG and IgM. Okay, so whenever IgG or IgM will be produced, it will bind to a bacteria, it will result in activation of the classical pathway. Then if you look at number 2, the name tells you a lot about the activation. It is called as mannose binding lectin. Do you all know what is mannose? Mannose is a carbohydrate. Very good. So mannose binding lectin. So mannose carbohydrate present in the bacterial wall, it will be bound by the lectin, and the mannose binding lectin pathway is activated. Finally, the alternative pathway, there are certain bacteria that are encapsulated. So they have a capsule around them, and it's made up of polysaccharides. So encapsulated bacteria are going to activate the alternative pathway, and then we'll see what happens to that. Before going into the details, there are certain basic things that I'll write down here about the complement proteins, and then we'll look into the details of each pathway. So one thing is that there, I've just written like C1 to C40. So these are just to tell you that there are over 40 proteins that are part of the complement system. I have written here only 5 of them: C1, C2, C3, C4 and C5, because they have certain important features. C1 has got 3 components: a Q chain, R chain and an S chain. That's why it is written as C1QRS. Then C2, C3, C4 and C5, they are normally inactive in the blood, but whenever they are activated, they will be converted into 2 parts, A and B. So C2A, C2B, C3A, C3B, C4A, C5B. So each part will be converted into 2. Then the remaining 6, 7, 8 and 9, they are just as such, they will become part of the complex. We'll see how it happens. Now we'll start with the classical pathway. First, so the classical pathway is going to start when the bacteria enters the body. Immunoglobulin G and IgM, they are going to bind and make antigen antibody complex. Okay, so that's the starting point of the classical pathway. Immune complex formed between IgG, IgM, any of these. Mostly it is IgM is more stronger than IgG, and they bind to it and they will fix. So there is a word you will find in the book, complement fixation. So what does that complement fixation means? That C1QRS, it is in the bloodstream. Whenever this immune complex is formed, they are going to bring that C1 and fix it on the bacteria. Okay? Everyone is okay with this one? That fixation of complement system means that antigen antibody complexes will activate C1, makes it fix, bind to the wall of the bacteria. So now the C1 is bound here, QRS, first Q will be activated, then R and S will activate C4 into which components? C4A and C4B. C4B will become part of the classical pathway. What happens to A, we will see later. Then the C4B is going to activate C2 into which components? C2A and C2B. So C4 will be joined by C2A. Which number is missing so far? No, it started with 1. gone to 2 and 4. Any number missing in between? 3. So C4 and C2A, they collectively are called as C3 convertase. What does convertase means? Convertase. What does convertase means? To convert. The enzyme that is going to convert C3 into C3A, C3B. So now this group become normal. So it's just simple numerics. 2, 3 and 4, they are all joined together. What's the next number? Yes, this group together, C4B, C2A, C3B, this is called as C5 convertase. And what's the next number? 6, 7, 8 and 9, they all will join in, just simply. When C5B, C6, C7, C8 and C9, they join together, this is called as Macintosh, no? It's membrane attack complex. Okay, so this final thing is called as membrane attack complex. You might have noticed that we have not used C3A, C4A, C5A. They were produced, but we kept them on site. What is their function? We'll talk about that. So I'll just briefly explain this again. There are 3 pathways: the classical pathway, alternate pathway, mannose binding lectin pathway. The classical pathway is activated whenever antigen-antibody complexes are made. Between which antibody? IgM and IgG. When they bind together, they are going to first activate C1q. And it has 2 other components, R and S. They will activate C4 into A and B. The C4B will join, and then that will activate C2 into A and B. C2A will join it. So C4B and C2A together is called as C3 convertase. That will convert C3 into A and B. So now 2, 3 and 4, simple. Just if you remember 1, 2, 3, 4. The numbers are coming. So 2, 3 and 4 together are called as C5 convertase, which will again convert C5 into A and B. C5B, C6, 7, 8 and 9, they join together to form MAC, which is called as membrane attack complex. Then C3A, C4A, C5A, they also have very important function, but at the moment just you remember that they are called as anaphylotoxins. I'll explain what is that in a moment. Then we come to the second pathway, which is the mannose binding lectin pathway. So it's very simple to remember. It's exactly the same as classical pathway. Okay? The only difference is the starting point. So as I told you already, mannose containing bacteria will activate this pathway, and there is no C1, just there is another protein here which is mannose activating serine proteases. So you don't need to remember that, just this replaces C1. So the Difference is in starting point and this one protein. Rest of it is same. C3 convertase, C5 convertase, MAC formation. Okay. Now the last pathway is called as alternative pathway. But before we go into that, you can see first the two columns. Classical pathway has got initiating protein as C1. The mannose binding lectin has got MASP1. So it's just the starting protein. And the alternate pathway is C3. Now it's mentioned which conditions, but don't get confused here. We'll go first to the animation. We'll see all the steps that we saw here. So this is a bacteria, okay? You can see the wall of a bacteria. What will happen in the classical pathway? Which antibodies should come first? IgG and IgM. So here comes IgG and it binds to the bacteria and it activates C1q. C1q will activate or change C4 into C4b and also C2 into C2a and b. This will now make C3 convertase. So far everyone is clear about it? Then C3 convertase is going to convert C3 into A and B. Now what this should be called? C5 convertase. And now this small block is explaining about the A components. Don't worry about it, I'll explain it. We'll go next what happens further. So this C5 convertase is going to convert C5 into A and B. 5 comes to the wall of the bacteria and then it is joined by others 6, 7, 8 and 9. And what is it called? MAC, membrane attack complex. And how it destroy the bacteria? It punch hole in the bacteria. So you can see this cylindrical C9 is basically like a cylinder that is punched into the wall of the bacteria, and this is the electron micrograph of a bacterial wall. So you can see made holes in the bacteria. Now what will happen? Electrolyte, will come out of the cell, the fluid will rush into the bacteria, and as a result the bacteria is going to be lysed, it is going to be destroyed. So this was classical pathway. Mannose binding lectin pathway is exactly the same. The only difference is the activation is by antigen and antibody or not? No. So mannose binding lectin pathway is second group. This one. What activates the mannose binding lectin pathway? Mannose present on the bacteria. Is antibody involved in the second pathway? No. And as a result again MAC will be formed and C3a, C4a and C5a will be formed. And this is the example of a gram-negative bacteria that was destroyed by the complement protein. Now the alternative pathway, it's different from the classical pathway. Once again, the antibodies are not involved. Just remember this thing, C3, it is normally in the body converted into active, and then if within microseconds if does not find the bacteria, it's going to become inactive. So like it's a cycle going on. C3 become active and then inactive, active and then inactive. So it's constantly happening, but whenever the bacteria come and C3 finds polysaccharides, encapsulated polysaccharide, then it binds to the bacteria. Okay, and then the alternative pathway or the alternative pathway will begin, or it will start. So C3 is normally in the blood, becoming active, inactive. It's going through this spontaneous hydrolysis, and whenever the polysaccharides capsule encapsulated bacteria will enter your body, it will directly bind to it. Otherwise it will get inactivated. After that, what happens? So the first thing is C3b. So here's the bacteria. It finds C3b in active form. Then there is a different protein here which I didn't mention here. This is also a complement protein. It will bind to this and they will again form a C3 convertase. So the C3 convertase of alternate path is slightly different from classical and mannose binding lectin. What should this C3 convertase do? What should it be doing? C3 convertase. It will convert more C3 into A and B. So it will be joined by more C3B. And now this will be called as C5 convertase. And then C5B, C6, C7, C8, 9. What is it called? MAC. So now I'll just explain once again the initiation, the classical pathway initiated by antigen-antibody complexes. Which antibodies? IgM and IgG. First thing happens is C1 is activated. That C1 will then activate C4, C2, that they will make C3 convertase, then it will make C5 convertase, finally C5b, 6, 7, 8 and 9 will MAC formation will occur. Mannose binding lectin pathway exactly the same. The difference is mannose of bacteria and MASP1 that will activate it. Other C3 convertase, C5 convertase, MAC formation is exactly the same as classical pathway. Third pathway is alternate pathway. C3 is going into activation, deactivation, spontaneous hydrolysis, but whenever encapsulated bacteria that comes in the body, it will bind to it, and C3b will find a capital B and a small b, make C3 convertase, make C5 convertase. As a result, MAC formation will take place. And in this pathway also, C3a, C4a, C5a will be produced. We'll see what are the functions. So all of you are so far okay with the activation, three main steps: C3 convertase, C5 convertase and MAC formation. Everyone is clear? Yeah. Now this is the animation for the alternate pathway. So you can see spontaneously hydrolyzed C3 will come. It will activate into C3b, bind to the bacteria, polysaccharide of the bacteria. As a result, it will be further joined by factor B or complement protein B. It will change into capital B, small b, BA will move, and this will be called as C3 convertase. C3 convertase will convert more C3 into A and B. Now it will be called as C5 convertase. C5b will join the bacterial wall, 6, 7, 8 and 9. Again, MAC formation and holes will be made in the bacterial wall.
Where does CA go?
All of it is in the blood. They have so now the complement proteins have lot of functions. We'll see how those functions are met. The MAC formation is doing the function of destroying the bacteria, and C3a, C4a, C5a, they are performing the function of anaphylatoxins. We'll discuss that just a couple of slides later. So this is once again the classical pathway, antigen and antibody complex. Mannose binding lectin pathway is activated by mannose of the pathogen. Alternate pathways by the encapsulated polysaccharides. You can see classical pathway has C1q, R and S, C4 and C2 involved. Mannose binding lectin pathway has MASP1, and the alternate pathway has C3. So if you want to discuss the differences, classical starts from 1 till 9, alternate starts with MASP1, so C1 is not involved. 2, 3, 4, 5, 6, 7, 8, 9 all are involved. Alternate pathway 1 and 2 are not involved. From 3 onwards everything is involved. Okay. We have seen the formation of MAC here, the terminal component, and MAC will destroy the bacteria, this one. But the remaining functions, as your colleague asked, what about C3a, C4a? They will perform some other functions which are discussed now in the next slide. So first thing is, what about C3a, C4a, C5a? So from the letter A, you should remember anaphylaxis. Have you heard of the word anaphylaxis?
Yes.
Yes. What are the symptoms of anaphylaxis?
Swelling. Edema.
Very good. Swelling and edema of bradycardia. Excellent. Why bradycardia occurs? Vasodilation of blood vessels. So what are the features? First thing is your bronchioles constrict, so breathing is labored. Okay, so when the bronchioles constrict, the patient cannot breathe. That's one. Then there is increased permeability, so the mucus of the respiratory tract become edematous. It is swollen. Again, the narrowing of the respiratory tract. The vessels, you talked about the blood vessels, they will dilate. So the peripheral vessels, when dilate, because of gravity, hypovolemic shock. Okay, so because the peripheral vessels in your feet, in your legs, they will dilate, hypovolemic shock. So anaphylaxis is a mixture of all these things. There will be increased permeability of the blood vessels, there will be increased permeability of the epithelium, there will be bronchoconstriction, smooth muscle constriction, and vascular dilatation. All these features of anaphylaxis are also produced by anaphylotoxins C3A, C4A and C5A. You can see now in this figure that C3A, C4A, C5A, they increase the permeability of the epithelium. Then they call more immune cells towards the site of inflammation. So wherever this complement protein is activated, they will call more immune cells there, and they will release histamine, bradykinin, which will perform the function of vasodilatation, bronchoconstriction, vascular dilatation. So all the features are as a result of these three, C3A, C4A, C5A, which triggers histamine release, bradykinin release, and increase in permeability, vascular dilatation, smooth muscle constriction. So one function of the complement we saw is MAC formation. Another function of the complement is anaphylotoxin, as an anaphylotoxin, whether increase in permeability, vascular dilatation, bronchoconstriction or smooth muscle constriction, and chemotaxis, calling other immune cells. Then it has another function which is called as opsonization. Have you heard of opsonization? Very good. Opsonization is called as simply tagging. So a bacteria come, if I tag it like this, now the macrophages, monocytes, neutrophils, they can recognize it. So whenever the tag of an antibody or a complement protein is on top of a bacteria, the other immune cells will recognize it and engulf it. So here in this figure you can see C3b, it is bound to the bacteria. Okay, so this means it is opsonized. And in the next you can see green macrophage, it can recognize it with the help of CR1, which is complement receptor 1. Once it recognizes it, it is going to eat it up, engulf it. So opsonins help in phagocytosis. Okay, complement protein will tag the bacteria so that macrophages or monocytes can recognize it, and then they are going to engulf it, eat it. So opsonization helps in phagocytosis. Is it clear to everyone? No. Does it make sense, or because a lot of faces seems to be lost somewhere? You can ask the question, because I always feel any session or any lecture is the best time for you to make questions, because close to the exams you lose that opportunity. Then is our time to ask the questions. So you can ask whatever you like. If you have not understood something, just raise your hand and I'll explain.